Hello, and thank you so much for joining my presentation. My name is Patrick Langley. I am a dietetic technician registered, and this is my capstone project presentation for the University of New England. The title of this project is The Role of the Microbiome in Patients with Crohn's Disease as Measured by C-reactive protein, a Systematic Review. And I just wanted to point out that all photos in this presentation are provided courtesy of Pixabay as indicated. So as part of the introduction, what is Crohn's disease? Crohn's disease is one of the two types of inflammatory bowel disease that affects the gut health overall. The immune system and other factors actually contribute to the increase in bodily inflammation, and this inflammation leads to what is known as Crohn's disease. The differences between Crohn's disease and ulcerative colitis, the other form of inflammatory bowel disease, is that ulcerative colitis is only specific to the colon of the body or the large intestine, and Crohn's disease measures from the mouth to the anus. Crohn's disease is also measured by a measurement called Crohn's Disease Activity Index. This index is utilized to determine the overall presence of the disease because it is multifactorial, and therefore the disease requires more than one or two measurements of disease-related activity. So what is the microbiome? This picture in the left will be our great demonstration for the microbiome, as we would like to think of microbiome or probiotics as being our friends that live inside of us and are able to help us with improving our overall health. Furthermore, as previously mentioned, what are probiotics? Probiotics are a portion of the microbiome that increase the health effects of the gut. Therefore, they help in decreasing overall inflammation in the gut, some of them, and they also provide other benefits such as production of lactose and the digestion of different food components to improve overall digestion, health, and they contribute to many other factors in health. It is not just the gut that probiotics help. The objective or the goals of this study are for you to gain a deeper understanding of what Crohn's disease is, its causes, and other effects that can help in improving the overall disease state. The purpose also of this presentation is to examine the results of this systematic review, explore probiotic supplementation and its benefits in Crohn's disease specifically, and compare the microbiome of Crohn's disease patients to that of healthy controls to determine how these probiotics help or don't help the body in the onset of Crohn's disease. In background, Crohn's disease in the 20th century was very rare. It was not a very common disease at this time, and its occurrence was only treated with medicinal interventions. The food system and its increase in processed foods and thereby improving the amount of cheap food available to people made it easy for Crohn's disease and ulcerative colitis to become present in today's population. And this is not just limited to the United States or Europe. It is rampant in many different parts of the world. The Western diet, also high in processed food and sugar, has contributed to this inflammation, as these foods in particular are known as being pro-inflammatory because they are high in omega-6 fatty acids and sugar. They should be considered for the dietary components of those with Crohn's disease. However, this particular study did not take that into account, and we will talk more about that as the presentation continues. The inflammatory lab markers that are taken into consideration in this study are C-reactive protein and other inflammatory markers that studies consider for their study outcomes. the microbiome is not just probiotics. It does include some pathogenic bacteria, and they play a role as well because they increase the odds and the outcomes of those with Crohn's disease. Short-chain fatty acids are mentioned in this slide because some of the probiotics specifically associated with Crohn's disease and better outcomes are those that produce short-chain fatty acids from the fermentation of fiber in the body. And this actually helps to decrease the overall bodily inflammation found within the gut. Probiotics that are specific to this concern are Faecobacterium prausnitzii, and this particular probiotic will be mentioned further throughout the presentation. The research question of this study is, among adults greater than 18 years old, how does the microbiome affect inflammation of patients diagnosed with Crohn's disease as measured by C-reactive protein? Part 1 of the methodology goes over the search strategy. The search strategy used for this study included Boolean phrases and other phrases that helped in providing a comprehensive findings of different studies and different study designs that met the research question. The search databases utilized were PubMed, PubMed Central, and ProQuest Central. The search terms that were used included Crohn's disease, the letters CD, and older adults, microbiome, microbiota. It also included and it also included the words adults and other terms that were related to the research question. The methodology part 2 goes over the inclusion and exclusion criteria considered for this study. The inclusion criteria required that the articles be within the English language and the proper age of greater than 18 years old, and there was no maximum age considered for this study. However, majority of the people contributing to the study were between the ages of 30 and 70 years old throughout all 16 of the articles chosen for the study. The publishing time frame was defined as between September 2009 and September 2019. Any articles published before that time frame were not considered. The inclusion criteria also considers the need for providing probiotics as an intervention for those specifically with Crohn's disease, or that they were comparing Crohn's disease and healthy controls. All of these were considered for inclusion and exclusion criteria. Other exclusion criteria not mentioned include diagnoses such as chronic heart failure, type 2 diabetes, and other metabolic or health or nutrition-related diseases that would affect dietary or inflammatory-related outcomes due to their high inflammatory nature. Each of the 4 studies were classified into 4 different categories of either A, B, C, or D based upon their strength. This was considered by the Evidence Analysis Manual of the Academy of Nutrition and Dietetics, and this manual was utilized to provide the best nutrition-based formulation of systematic reviews within this field. The risk of bias was considered throughout the entire study because of the selection of the articles and different considerations throughout the entire study. The primary outcomes were C-reactive protein and other variations of the microbiome when the studies were comparing the microbiome of healthy controls and Crohn's disease patients. The total number of present bacteria were compared to consider if they were significantly variant. A total of 591 articles were initially found, and they were considered based on their titles. After that, the articles were excluded after further reading and consideration of whether they met the criteria of inclusion and exclusion for the study, and therefore 81 of the 97 articles that were considered for full review were excluded, and only 16 of the studies were included in this systematic review. Other studies were utilized to compare outcomes of other systematic reviews and to add for considerations of other factors within the study. In the results, Part 1, considerations for different strains of probiotics and probiotic-rich yogurt were utilized to determine outcomes. The purpose of providing these probiotics was to see if they were able to increase the presence of gut microbiome in patients diagnosed with Crohn's disease. The increase in these particular probiotics would provide benefit to the host, especially when diagnosed with Crohn's disease. Results, Part 2, the location of different presence of Crohn's disease was considered, such as ileocecal valve, ileum, and anus, anal fissures, etc. All of these different locations were considered in the comparison of the present microbiome and compared to healthy controls. The majority of these studies determined a significant variation in the microbiome between healthy controls and Crohn's disease patients. In the patients with Crohn's disease, they actually had an increased level of pathogenic bacteria and a decreased level of probiotic bacteria that affected their overall health and inflammatory status. For example, in all of the studies, they found that the bacteria, Fecal Bacterium prausnitzii, that has shown to decrease overall inflammation by fermenting prebiotic fiber into short-chain fatty acids, were actually depleted significantly in patients with Crohn's disease, and they were higher in those of healthy controls. Here is a visual abstract of the study and its associated outcomes in the groups where Crohn's disease patients were compared with healthy controls. They actually saw, as previously mentioned, a decrease in Fecal Bacterium prausnitzii bacteria that was considered a functional strain in Crohn's disease. And as you can see in this graph, as the Crohn's disease activity index increased this direction, there was a substantial decrease in the microbial dysbiosis index, which means that participants with a higher level of Crohn's disease activity had an increased chance of microbiome dysbiosis, which indicates that the microbiome has an essential role to those with Crohn's disease and others in the prevention of associated inflammation in the gut. Moreover, in the probiotic supplementation group, it showed that when patients consumed high levels of probiotics and the probiotic-rich yogurt, they saw an increase in the overall microbiome. And this was, as previously mentioned, the demonstration of our lovely microbiome that is our friend and helps in improving the health of our bodies. And it also saw that a decrease in the total pathogenic bacteria saw a relatively lower level of lab values that included C-reactive protein and other inflammatory markers, Crohn's disease activity index, and a decrease in pathogenic bacteria when probiotic supplementation was provided. In conclusion, probiotics in the microbiome are an essential portion to the prevention and management of Crohn's disease and other inflammatory bowel diseases. Those with active Crohn's disease would benefit from probiotic therapy to reintroduce necessary bacteria. Due to the varying probiotic strains and different factors that affect overall health, this study receives a fair grade overall. and should be considered for future studies and research.